Space Center's director. We're very happy to have Garabet Kazanjian with us. Garabet has recently graduated from Humboldt University of Berlin in Limnology. And for some of us who never knew what Limnology was, that's the study of lakes, ecology. And he comes with quite an interesting background of working not only as a scientist and researcher for many, many years, going back to his undergrad and master's work also at AUB, American University of Beirut, but he also worked with Greenpeace on activism around creating marine protected areas. So he comes with this very interesting combination of both political policy work and also science. So with that, we're very much looking forward to your presentation. Thank you.
Thank you, Alan, for the nice introduction, and everybody else for showing up and expressing interest in the talk. Can we?
Yeah.
So I'm going to, I know not everybody here are basically is a limnologist or an aquatic expert. Perfect. I'll try to make the talk as general as possible, and if I use any word, any terminology which somebody doesn't understand, feel free to stop me and I can explain. Otherwise, there will be around 20-25 minutes at the end for questions and discussions. So let's start. Okay. We start with one of our neighbors from the region. He actually lived in what nowadays is Turkey, Heraclitus. He was quoted by Plato to have said, "Everything flows, and nothing stands still." He's also known to have said, "No man enters the same river twice." So, in my mind, he seems to have been one of the world's earliest aquatic ecologists, at least by heart, if not by actual profession. But all jokes aside, the main point of his message is that change is the only true constant in life. And while that may be true, our planet has seen an unprecedented, maybe the colors are not so clear, it's seen an unprecedented wave of change, at a very fast pace. So everything here you see in darker colors, or even these shades of lighter red, they're basically the regions which registered a higher or a warmer climate than their average climate regime last year. This is the data from 2019. So as you can see, it's pretty much all over the globe except for very few bits and pieces. And including the region where we are now, it's basically expected to get warmer and warmer. So what does this mean to freshwater ecology? What does this mean to the lakes, to the rivers, and why should we care? So if you had asked me that...
Might want to use your finger to do so for me.
If you had asked me that question maybe a couple of decades ago, I would have said not much. The way it was seen that freshwater systems, lakes and rivers, were just a passive funnel, a pipe. Basically, all the carbon gets synthesized, sequestered by the plants, by the primary producers in terrestrial ecosystems, and then whatever dies gets transported through the rivers, through the lakes, and ends up in the ocean. So it was just a transport mechanism. It was not active, it was passive. This started changing a couple of decades ago, or around the late 80s, 90s. Because when they started adding up the numbers, it didn't make sense. So they added basically how much carbon leaves the terrestrial ecosystems and how much of it ends up in the ocean, and it was only 1/3. So what happens to the rest of the carbon? Where does it disappear? Where does it go? And then they started looking into the transport mechanism. What happens there? Oops. So then they saw that part of this carbon, which goes into the into the freshwater ecosystem, gets transported back to the atmosphere. It gets emitted as carbon dioxide, CO2 or methane, CH4. And the rest of it gets buried into the sediments, basically gets stored there. And whatever remains ends up in the marine ecosystems. So this is a way which we use to classify whether these systems are carbon sinks or sources. If they emit more carbon back to the atmosphere, then they're labeled as a carbon source. They contribute more to climate change, to greenhouse gases. If most of it gets buried, it's a carbon sink. It's a good thing, because they're taking this carbon from the surrounding, from the landscape, and basically burying it, putting it in a bank of sorts in the soil. So, naturally, when the researchers understood that freshwater ecosystems are important, they started looking at the big lakes, at the larger lakes. Because, well, they're bigger, they contribute more, obviously, and they're more important. But that's not necessarily true, because while, if we look at one lake versus one pond, for instance, the bigger lake has, on a singular level, a bigger surface area. But for each large lake, for each lake 7, you have a lot of smaller lakes. So if you add up all the numbers together, globally, they actually are very similar in total surface area. So basically you have small freshwater ecosystems which contribute to 1/3 medium-sized and large-sized lakes. So altogether, small freshwater ecosystems should not be neglected because they contribute equally to the larger counterparts. Furthermore, if we look into the emission rates from smaller ecosystems, the smaller the area, we have higher CO2 emissions and even more so higher methane emissions coming from smaller ponds per surface area versus the larger lakes. So we established that freshwaters are important.
Sorry, can I interrupt one second? This includes reservoirs? Or
yes,
okay.
yes. So we basically settled the fact that freshwaters are important for carbon cycling and small freshwaters are also important. Furthermore, in smaller systems, we have the benthic component, which is extremely important because the areas are shallow, there's a lot of light penetration, so a lot of plants grow, a lot of benthic algae, periphyton grow as well. So they contribute a lot to primary production, to carbon sequestration. But again, because the trend was to look into larger lakes in the beginning, the majority of the papers that came, well, this is a bit old now, it's 2 decades old almost, but still, the overall trend was that the majority of the studies focus on pelagic production, on phytoplankton, while they actually ignore the benthic, or basically studies that combine both. So that's another aspect that we should focus on, the benthic production, because it's also important. Altogether, I took this, plus the modern factors or problems that we have in the world at the moment, and I tried to study how these affect freshwater ecosystems and carbon cycling in freshwater ecosystems. So I saw that there's a lot of nutrients being dumped into freshwater systems by urban projects, development, etc., and also from agricultural fields, for instance. There are a lot of water level changes happening, either due to urban developments, drastic weather conditions, etc., etc. The organic carbon, dissolved organic carbon is increasing. I'm going to get to that in detail further on in the presentation. And of course, the big elephant in the room is global warming. So how are these affecting freshwater ecosystems? I'm going to talk about 3 studies I've made in the last few years, which actually combine these components and their effects on these systems. So study 1 combines the first 2, second study combines the second 2, and then the last study is about future predictions with regards to warming up the planet. So, to focus on the first study, it was part of a big project actually. It was called Landscales. I was working at IGB, the Leibniz Institute of Freshwater Ecology in Berlin, and we collaborated with another German institute called SALT, which is a more terrestrial agricultural institute. So we wanted to find the link between the terrestrial carbon, the terrestrial nutrients that were coming in and going into the water systems, and basically what was happening to it. Where was the carbon ending up? What was happening to the nutrients? So to do that, we wanted to focus on very small systems, which got a lot of carbon and a lot of nutrients from the surrounding. So we ended up in this region, the north of Germany. This is Berlin. This is around 200 kilometers north of Berlin, close to the Polish border, where there are a lot of small ponds, which they actually call kettle holes, or in North America, they're known as potholes. So I don't know if you can see in the picture, basically there are small ponds located in agricultural areas, in massive agricultural fields. There are around 300,000 of them in northeastern Germany. It's around 5% of the total arable land. And we don't know anything about them because aquatic scientists think they're so small, they never study them. Terrestrial ecologists think these are water, we don't deal with them. So they're kind of in between the two ecosystems and they're neglected. So we don't have any data on it. They get massive flow of nutrients from all the fields, from fertilizers and so on. And because they're small, they fluctuate a lot. So if it's a hot summer, the water level drops down. If it's a wet summer, they float. And we wanted to see what happens overall. We focused on two common types of these kettle holes or potholes. One had a mixed vegetation, a lot of different aquatic plants, macrophytes, and the other one had a surface cover of duckweed. So it was completely covered with duckweed. It had a reed belt around it, but basically light couldn't penetrate in because basically it got reflected off the duckweed on the surface. So what did we do? The most common or the easiest methodology to use to basically estimate primary production and ecosystem metabolism is to put an oxygen probe in the middle of a pond or a lake. And then you measure the oxygen every 30 minutes or every 1 hour. And you basically get some kind of a sinusoidal curve like this. So during the day, because of photosynthesis, oxygen increases. During the night, there's no photosynthesis, all the plants are respiring. You get a decrease in oxygen. And then you start to add up all the increase, that's your total production. The decrease is respiration. You subtract one from the other. If it's positive, then you have more production than respiration, so it's a carbon sink. If there's more respiration, then it's a carbon source. However, we tried to implement this system in those ponds. It didn't work because then there was a big long periods throughout the year where we didn't have enough oxygen in the ponds. So what happens if you put an oxygen probe to measure oxygen and there's none? You can't use the methodology. So we had to look for another possibility. We went for a compartmental approach, which was basically to measure all the algae in the water, the phytoplankton separately. We measured all the periphyton, everything which was growing on the benthos, on the sediments. We put plastic strips, we incubated them for a month every time, and then we collected the strips and measured how much production there is, how much algae growth there is. And then we also measured all the macrophytes which were in the ponds. Together we summed up all these production figures, and then we have the gross primary production of the lake. Furthermore, we studied the water chemistry to see what's happening to the nutrients, sediment mineralization rates, or decomposition rates, sediment deposition, how much of this algae, the plants, was actually sedimenting to the bottom, and we tried to measure carbon dioxide and methane emissions as well, although that wasn't a very successful part of this project. So the main hypothesis or questions we had was, how much do we actually have production in these systems, gross primary production, and how does it compare to other systems, to bigger lakes, for instance, because we have no data on this at all worldwide. What was the different contribution of the different primary producers? So since we actually took the effort of measuring each single one, the phytoplankton, the periphyton, and the macrophytes, we wanted to see how it compares and which one contributes most. And then how do these primary producers, how does the whole thing impact the carbon cycling dynamics? So these are the production rates, the figures. And the first thing you notice, so these are the different components in different colors. These are macrophytes and periphyton and phytoplankton. The first thing you notice is macrophytes basically have the majority of the production. And it mostly happened during the spring-summer months, where you had the macrophyte growing season. The rest of the year, it was the periphyton contributing to a bit of the production, and there was no phytoplankton. The main reason being was we had no fish in those kettle holes, because sometimes they dry up and the fishes die. So in the absence of fish, we had very high zooplankton, and the zooplankton were almost always grazing whatever the phytoplankton was appearing. So that's why the phytoplankton levels were very low. We had a bit of periphyton, but the majority of the production was going into the macrophytes. This is basically a confirmation of what I just said. In one kettle hole, the mixed vegetation, 90% of the production came from macrophytes, In the other one, 81% of the production also came from the plants. And this is how the kettle holes compare versus other ecosystems, so freshwater ecosystems. So if we take the annual production rates of other ecosystems, we see that kettle holes are actually on the higher end of production rates. They have a very high production. If we take only summer rates, then they're actually even higher than most of the other aquatic systems. So we established that there's a lot of production. What happens to the carbon which is being sequestered, which is being synthesized? We saw that GPP is high. We saw that there's a good trend with GPP, with production in sediment deposition, especially in the second kettle hole with the floating vegetation. But we also noticed that mineralization rates, decomposition rates, or aerobic decomposition rates were extremely low. And that's basically because there was no oxygen. Usually if you have aerobic mineralization, you need oxygen to break down the organic matter into smaller elements. But since there was no oxygen in these kettle holes, or very little oxygen, mineralization rates remained very low. So it means, basically, the conclusion of the story was we had very high production. A lot of this production was actually sedimenting to the bottom of the ponds, but it wasn't being mineralized, which means it's likely going to be stored. So these kettle holes have a high potential of storing carbon, of functioning as carbon sinks, as long as they're not disturbed. The twist in the story is, once in a while, because these kettle holes are not very deep and they're not very big, if we have very dry summers and very hot summers, they dry up. And once it happens, or you get a very low, basically, depth of water, the sediment gets oxygenated, gets aerated. And then you get basically all the carbon which was stored gets mineralized again and gets re-emitted to the atmosphere. Actually, this is what happened probably also in Armenia. I know from Eastern Germany, for instance, during the communist era, they took a lot of the wetlands, a lot of the peatlands which were immersed in water and were anaerobic, and they were basically collecting a lot of the, storing a lot of the carbon for decades and centuries. To be self-sufficient in the agricultural sector, they dried up a lot of these wetlands and turned them into agricultural fields. But what happened in the meantime was when they dried up these peat, soil, a lot of the carbon which was stored for a very long time got emitted to the atmosphere. So in terms of, okay, before I end up in the conclusion, another point, how the primary producers affect the carbon cycling. We saw, especially in the second kettle hole with the floating vegetation, that when oxygen rates, so this is the oxygen concentration throughout the year, and we see it's very little, it's almost anoxic throughout the year. And whenever we had anoxia, we had a very high concentration of phosphorus in the water. And this is basically phosphorus being released from the sediment in anoxic conditions. Whenever we had oxygen appearing in the system, you have all these redox reactions, and phosphorus sediments back into the sediment. However, we couldn't have a lot of oxygen because we had duckweed on the surface. So when there's no light penetration, all the algae dies. When the algae dies, you lose your oxygen, you have a lot of phosphorus released into the water, and duckweed lost phosphorus. So in a way, this is a feedback loop. Duckweed creates anoxia, anoxia creates phosphorus release, phosphorus is taken up by duckweed, which is a very phosphorus-loving organism, and it needs a lot of phosphorus to propagate. So as long as you have this feedback loop, the system is kind of stable. It keeps on staying in the same loop, basically, and repeating itself. So the conclusions of this study were that kettle holes are extremely important when it comes to production. The macrophytes, in our case, contributed to most of the production. The rest of the year, especially in the summer, the rest of the year we had periphyton production taking over. And that primary producers played a very dominant role in terms of nutrient cycling, oxygen concentration or availability, and carbon dynamics. Now I want to step back and see how this reflects back on ecosystems in Armenia. I know Armenia has, if I'm not mistaken, 3 Ramsar protected sites of wetlands, including this one, which is the Khor Virab Marsh. which has very similar kind of landscape. A lot of potholes, a lot of stagnant water in the ecosystem, a lot of agriculture around. So basically these systems, I haven't studied, but I think they're very comparable. And these systems can be very important in storing carbon. They have to be monitored, they have to be protected, because we've already realized that once they're disturbed, whatever they stored, they can release back to the atmosphere in greenhouse gas emissions. And we need to basically find a long-term planning strategy to see how much of this carbon we can store into these ecosystems. So I moved to the second study. It was called the Theralac Project. It was about a lake, which was already eutrophic. And then around 2012, there was a lot of rainfall in the region. So there were floods ending up with the lake level rising. With the lake water rise, we had a lot of carbon moving from surrounding peatlands, from forested areas into the lake. And this is actually not specific to this lake. We've seen this scenario happening in a lot of different places in Central Europe and in the Northern Hemisphere in general. As you can see from this graph, a lot of the lakes, so around 24%, have seen an increase in DOC concentrations, dissolved organic carbon, from the 90s onwards. And once the DOC concentration in the water increases, the water gets browner and browner. So you can see this is almost no DOC, and then with an increase in DOC concentration, this is what becomes of the water within the lake. So when this happens, when the water becomes browner, less light penetrates into the water column, and hence the thermocline starts raising up. The thermocline is the level where the water stops getting mixed. So you have mixing here, and then here it's separated because it's much colder, so the water is separated. But when there's less light penetrating, this thermocline increases. So you get only production here in this top part, but all throughout the bottom, it's probably anoxic and dead. So to study this, we basically went to this lake, which, as I said, around 2012, it had a very high precipitation in the region, and the water level increased. So here you can see, this is the water level. It was around, 50 centimeters or so, where the growth was, and then it started increasing. With the water level increase, the DOC in the lake also started increasing. It went up to 5-fold. It wasn't just the DOC, also nutrients, total phosphorus increased as well in the lake. So I joined this institute, and they were already working on this project. But then I joined in around 2013, so exactly at the peak of this brownification event. And I wanted to see if we give the lake enough time, a couple of years, what happens? Will it return back if the precipitation levels become normal again? Will it go back to its initial condition, the baseline condition? Or will it somehow stay higher up or in between? So what happened was, when the lake water went up, a lot of the trees around the lake also got flooded and started leaking even more carbon into the surrounding. This was the initial results. So gross pelagic production increased because there was more nutrients, more nitrogen, more phosphorus in the water. So the phytoplankton ended up basically with a higher production rate. But what happened for the benthic production with the periphyton is it decreased because there was no light penetration anymore. So as you can see, the water level after 2012 started decreasing again. It didn't go back, well, almost, slightly higher up than the baseline condition. So I wanted to see what happens if the DOC concentration will also go back down, same as the phosphorus. Will the benthic production recover if we get back to the baseline conditions? And how will these changes overall affect the primary production and carbon sequestration in the lake?
Are you taking bets?
Should I? Let's do that. What do you say?
I think it doesn't go down. That would be my guess. Concentration goes higher.
Any other guesses?
Wild guess.
Well, not entirely incorrect. So the concentration goes down after 2012, but it doesn't go back all the way down. So this was the baseline condition. And it somehow DOC levels stayed double. So not fivefold, but only twice higher. Whereas TP concentration stayed even more so. So this was around, I think, 1.5, and this was double. Since the concentration didn't go back exactly like it was before, same thing happened with the light attenuation. So light conditions, this was the attenuation, which is basically how much of the light gets absorbed in the water column, increased when the DOC was very high. It started going back down, but it never reached the baseline level. And the euphotic zone, how much light penetrates into the water column, was 2.5 meters depth in the beginning, but then it increased, became very shallow, and ended up somewhere here as well. As a result, benthic production never recovered. Well, it recovered slightly, but it never went back. So this was the baseline production rate. It was around this high, 0.4 grams carbon per meter squared per day. And then once we got the peak brownification, it went almost zero. It started to recover in the last few years, in the next few years, but it got nowhere near the baseline condition. By the way, these results, the paper we tried to publish was, we capped it till 2015, but some colleagues of mine went and got several water samples from this same lake in 2019, this past summer, and it was still plateauing around the same concentration, so it hasn't changed till now. What we found very, very interesting was that we had an almost linear relationship from benthic production, between benthic production, DOC, and TP. So as DOC and nutrient concentrations increased, we had less and less benthic production. But this comes with a twist. The way we saw it was there was some kind of threshold line. When the organic carbon and the nutrient concentration increase further, then there's almost no light penetration. So benthic production goes almost to zero. Till that line, it's almost linear, but further on, and this is also basically seen in different mathematical models, you have this kind of threshold, which is around 30-35 milligram per liter DOC. Beyond that, you have almost no benthic production. So in conclusion, we basically saw that the water chemistry parameters never returned back to baseline conditions. Benthic production only recovered slightly, but not completely. And then these sudden brownification events might actually have long-term consequences. They might last for years or even perhaps decades. So how does this reflect to Armenia? The first thing that came to my mind was Lake Sevan. We know the lake has seen water level fluctuations for almost a century now. That's changing a lot of the water chemistry. We also know that the lake has increased nutrient and DOC loading coming from untreated sewage being dumped into the lake. We have aquaculture farms in the surrounding, which end up with more nutrients like nitrogen and phosphorus. And we have the same flooded trees phenomenon that we saw in the other lake in Germany, here as well, happening in Lake Sevan when the water level rose again after 2002. So how does this all affect the carbon cycling and the primary production in Lake Sevan? This is actually something I was discussing with Alan the past week, and we're trying to also come up with ideas to measure this in the lake. So this is from a paper from 2000 from Hovhannisyan and Gabrielian, which showed kind of the history of the lake. In the beginning, it had a high phosphorus concentration, but a very low nitrogen concentration. And the shift has already started in the 1960s when nitrogen concentration started increasing in the lake. And now nitrogen is somewhere around this concentration. So it kind of shifted from high phosphorus, low nitrogen to high nitrogen, low phosphorus. And that shifted also the ecosystem completely, where you had macrophyte or plant dominance in the beginning years, so before 1960s, and after 1960s, because the water quality decreased a lot, the plants mostly died, and then you had phytoplankton taking over and using the nutrients. And what happens when phytoplankton takes over is it shades even more the light because you have all the algae growing in the water, so they're also absorbing the light, and that basically ends up making it worse and worse for the plants. The other catastrophe is when you lose the plants, because they're taking a lot of the nutrients from the soil, they keep the nutrient level in the water column low. So it's kind of also another feedback loop. When the plants die, you get anoxic conditions. A lot of those nutrients get released into the water column, and then you also lose, or the water quality becomes worse and worse. So it propagates from a bad condition to worse and worse. And then when you have a very bad water quality, the plants will never come back. So in Lake Sevan, we've already seen a lot of this happen. We've seen the macrophytes have decreased. We've seen that the water quality has also decreased, it's becoming worse. And in the last few years, we've seen some algae blooms happening in the summer, which might lead to, if we don't fix it, it might go worse and worse. So at worst case scenario, it might become very similar to the lake that I showed in Germany. You have high eutrophication rates. You have no light penetration into the water column. You create an anoxic condition. All the fishes die. The lake starts stinking because it's releasing a lot of the methane and sulfur. And you have massive economic losses, both for fisheries, in terms of fish catches, crayfish, and so on. In terms of
recreation.
Yes, and ecotourism. So that was the second project. I'm going to move on to the third and last project, and this was about estimating the effects of warming on benthic and other primary producers. So we know global warming is happening, but we don't have conclusive data how this affects primary production. We know primary production increases with higher temperatures because cells multiply faster, but we don't know all the top-down and bottom-up effects that end up actually determining the production rates. So in the literature, we had some mixed results. Some papers said that production increases with increasing temperature, while other papers said that it actually shifts the controls, and you have higher top-down control, so it decreases production. So we wanted to see in a controlled environment what happens. For this, I was collaborating. I moved to the Netherlands for 2 years, and I was collaborating with the Dutch Institute of Ecology there, where they have 8 tanks of around 1,000 liter water volume. We ran the tanks, 4 of them in normal control conditions, like outside, and the other 4, we ran them at 4 degrees higher according to the IPCC scenarios of future warming. So we sampled phytoplankton, periphyton, zooplankton, and macroinvertebrates. And we basically saw, so the red is for the warmer treatment, blue is for the control. We saw that in the beginning, phytoplankton concentration unexpectedly were lower in the warmer treatment compared to the control. And this was because there was a fungal parasite infection that was stronger in the warmer treatment. It kind of killed the whole bloom much faster in the warmer one than in the control. And this led to more nutrient availability in the warm treatment because we had less phytoplankton. So we had a higher benthic production starting and an earlier benthic production in the warm treatment compared to the control. But we wanted to see if there was just this or there were other effects as well. So we measured the nutrient concentrations total in those tanks, and we saw that there was higher nutrient concentration in the worm already like a month and a half from the beginning of the experiment. And this we basically figured out that it's coming from the sediment. So although they started with the same nutrient concentrations, after a month and a half, we had higher concentrations in one treatment versus the other because mineralization rates, decomposition rates were higher in the warmer treatment. So they ended up basically pumping more nutrients into the water column, and this led to higher periphyton production in the water column. On the walls of the tanks. But then, if I go back 1 second, we see that higher periphyton or higher benthic production in the warm treatment occurred till this point. After that, it started decreasing sharply, and towards the end of the experiment, both treatments were very similar. So we wanted to see what happened at this stage. Why it started decreasing much faster in the warmer treatment compared to the control. So we looked into the grazing potential. We looked into the macroinvertebrates, the snails, for instance, the leeches and so on, and we looked at the zooplankton. So we see in both cases for zooplankton, in the warm treatment, the black one, they started coming up, appearing earlier and in higher abundance. In the case of the snails, it was the same. So the snails in the warm treatment were higher than the control. So on one hand, we had more nutrients, so higher production in the warm treatment. On the other hand, we also had stronger top-down control, stronger grazing. So it kind of, by the end of the experiment, it balanced out. And we had similar periphyton production towards the end. What did this mean to the overall carbon emissions from the system? So we calculated CO2 emissions and we also calculated methane. For the CO2, it was different in the beginning. It was higher in the warmer, but then after August, it switched. It was higher in the colder treatment. So at the end of the year, there was no significant difference. It was similar amounts of CO2 emissions from both treatments. But the bigger story was in methane. So with methane emissions, we have two types of emissions. One comes as diffusive from the water. It gets mixed in the water column, rises up slightly, and basically goes into atmospheric flux. And in this case, they were not significantly different. But we also have another different type of methane emission, which is by ebullition. It's basically bubbles forming in the sediment, and the bubble rises very quickly to the top and basically goes into the atmosphere. And when we measured ebullition, it was much higher in the warmer treatment than in the control. So overall, although CO2 is not different in both treatments, in the warmer treatment, when the climate was high, it had higher temperature, we had higher methane emission overall from the system. So to cap it all, what are the conclusions and expectations that we can take with us towards planning the future? We saw that the bottom-up effects actually had a positive effect on carbon sequestration. On one hand, we had higher and faster growth in production. On the other hand, we had more nutrients into the water column, so more carbon sequestration by the primary producers. On the other hand, we had top-down effects which had negative effects. We had higher grazing and earlier onset of grazing onto the primary producers. Because of this, the overall system production was significantly not different between the two treatments, so it balanced out, top-down and bottom-up. But the more crucial aspect to this is, as we saw, we have earlier onset of either blooms or grazing, so zooplankton, macroinvertebrates, and so on. And this is a very delicate matter in ecosystems, because once all these organisms start shifting their time periods when they appear or when they add up, it might start creating a lot of problems. So say the zooplankton appears when there's not enough algae, they won't survive, or vice versa. The other possible aspect is, for instance, periphyton production, benthic algae, start happening earlier in the summer, in the warm treatment. And this might pose a problem for the plants, the benthic plants, the macrophytes, which are only starting to grow around this period, around April to May, at least in Central Europe. So when the plants are still small and short, and you have benthic algae growing on top of them, and the benthic algae becomes so thick that light doesn't penetrate through it and doesn't reach the plants, it means the plants don't survive. And they might die before even reaching their natural height. And this will have, again, a lot of different consequences on carbon sequestration and water quality. And the final conclusion, as we saw, that we should expect higher greenhouse gas emissions, at least in this case with regards to methane, with warmer temperatures. So this was kind of the summary of 3 different research projects that I was involved in. Each project had a much wider scope, of course, linked to a lot of different work that you can see here in the publications. So we were working on different aspects of feedback loops and overall methane or CO2 emissions and bacterial processes in the water. But more importantly, for the sake of this talk, I want to see where we can go from here and also how we can implement it to Armenia. So on one hand, it's very important to start looking for less studied ecosystems, which might be important, similar to the first study where we analyzed the kettleways. Fill up the literature gaps, and then start to scale it up, basically over our landscapes to see how much carbon emissions we might expect now or in the future. Then we should also incorporate within our plans all the future changes in climate and extreme weather events, precipitation differences and so on, invasion of exotic species and how these could contribute to changes in carbon sequestration. And then according to all of these studies and conclusions, we have to find a way to improve our environmental policies and start implementing them to mitigate all the future environmental disasters that we might end up with. So I want to thank all the institutions that helped me and basically gave me the possibility to do these studies, as well as the funding agencies. And to end it all up, I want to, well, first of all, if you have questions about this talk, go ahead and ask. But at the end, I also want to get your feedback about what you think are problems in Armenia relevant to freshwater systems, some maybe interesting projects related to these research fields, and also ways that we can collaborate with each other. This is also my email address, so if you don't remember anything now and remember it later on, you can send me an email. You're welcome. With that, thanks again, everybody, for attending the talk. And if you have any questions, I'm all ears.
Okay, let me maybe start with one question. You showed that a chart from the study done in 2000, I think, where the nitrogen level, or the phosphorus level, which one it was. So there was a sharp kind of spike in the 1970s, I guess that coincides with the lowering of the kind of a culmination of the lowering of the lake as the Razdan Cascade was being built. Water levels were coming down. From 1930s on, you see
I have the graph from your presentation.
Right. So it's coming down. So by
this one?
Yeah, by 1970s, you are getting to the lowest point it has been. You suddenly see a spike, right?
Yes.
Which is, it'd be interesting to know why you get that sudden spike, as opposed to a gradual, increase. Second, are there, an interesting question, and maybe people who know the history of it might know, were there incidents of eutrophication back then because of this increased nitrogen and phosphorus, I believe was there also, concentrations? And if not, could it be that the climate change is actually playing a role in seeing more frequent incidents of eutrophication? Warming up,
Yeah. In terms of the spike, that's actually a very interesting question. So looking at it, it already starts increasing in the 60s, which is basically when the water dropped. And then all of a sudden, this spike that's coming up, it can be from three different actually explanations. One, we have less of a water volume, so less dilution from the nearby sources. Two, we might have an external event of nitrogen loading. I don't know what happens.
Agriculture,
yeah.
Heavy rains.
Could be. And three, and the other possible explanation is we have also internal loading. So if the water quality changed, and basically here it says end bottom sediment, so this probably is an internal loaded nitrogen. So if we have water quality change, the majority of the sediments, if the macrophytes started dying already here, the sediment turns anoxic, we might end up with a lot of nutrient release from the sediment, which were available or stored over decades or centuries even. So that might also explain this sudden. Usually, if you have a sudden change, it is likely that it's internal, unless you have a very strong incident that happened from external loading. If there's a specific event that we know that loaded a high quantity of nitrogen, then that might be the case. I don't know exactly what happened in the 1970s. You probably know better than me. But there's also a very high possibility that basically the water quality decreases gradually up to a certain threshold. Afterwards, the plants die, the sediment becomes anoxic, and you have this internal loop of all the stored nutrients being repumped into the water, especially if the water gets mixed. So that might be the case. And your second question was if climate change is making it worse.
Because algal blooms have become a phenomenon over the past 4 or 5 years. I mean, before that, how many incidents are recorded? In recent years, suddenly it becomes an issue. So, what could be the explanation for these repeated, frequent algal blooms?
So again, 2 cases. We know for sure for cyanobacteria blooms, they prefer warmer climates. And climate change is contributing to more and more cyanobacteria blooms worldwide. This isn't an Armenia only story. It's happening everywhere. So cyanobacteria blooms are becoming more frequent, mostly driven by warmer climates and also milder winters.
But the nutrient content matters, right?
For sure.
Yeah, okay.
So cyanobacteria like higher concentration of nutrients, which is why those type of blooms, you only get them in eutrophic lakes and not oligotrophic lakes, which have very low nutrient concentrations. So the water quality matters and the temperature matters. Those are the 2 main factors. In case of Sevan, I still have to look into the data to see how the water quality changed from that time till now. So it might be that the water quality is even decreasing further, and this is contributing. And then the warmer climates are an add-on, so to speak, of why they're making it more and more frequent or worse levels of cyanobacteria. So the climate we can't control to a large extent, at least if we're talking about Armenia, but the nutrient level we can control. So if you decrease the nutrient concentrations being pumped into the water, you can circumvent these type of blooms, even if the temperature still rises by a couple of minutes. Any other questions?
Question. So if I understood right, the kettle lakes were very efficient as carbon sinks, more so than their larger compatriots. If you were to, let's say, compare them to trees, if you took out a kettle lake and you just planted trees in their place, what is more efficient at sinking carbon from the atmosphere?
Depends on the trees,
Right.
depends on the forests. So for instance, a lot of the forests, rainforest type of things which have been storing carbon for decades or even more centuries, they're massive carbon sinks, right? That's why all the Amazon fires and whatnot are very important, because you're losing a lot of the carbon again. But in terms of a very high process, turnaround, these are quite efficient, because in terms of trees, they grow very fast in the beginning, but then they slow down. In the case of these kettle holes, they're basically growing every year, because in winter they're dying. And what happens is, when they die, they don't get decomposed easily, because then they fall into mud, where you have less oxygen, low mineralization rates, and it starts contributing to this, what eventually becomes peat soil, which is very high in carbon concentration. In the case of forests, it depends on the forest, so there's not one-size-fits-all answer to this.
Sure.
But in terms of these kettle holes or wetlands in general, they're very efficient in terms of constantly high rates of carbon sequestration, and then at the end of the day, most of it gets actually sedimented and usually basically as a carbon sink.
If I can follow up. Let's say if the wetlands dry out, how quickly do we lose the gains that we've made?
I don't have a precise answer on this, and actually not just me, it's still like we're only started to study these scenarios. But the most important answer depends on if it completely dries up or if you have still a bit of water left. So if the water levels drop really low, but you still have some water, where you can have all the bacterial mineralization happening in oxic condition, then it's faster. If it dries up completely, then of course you also kill a lot of the bacteria in the water, the aquatic bacteria at least, and then it happens slower comparatively, and it depends on the soil content. So if it's a compact soil and so on, how much oxygen penetrates into different layers, you probably get a lot of mineralization at the top layer of the sediment, but further below they might still be anoxic and not mineralize very quickly. So when we say dry up, we should probably separate between complete dry up, no water whatsoever, versus a very shallow layer of water remaining. In that case, it's probably worst case scenario because mineralization rates pick up, you still have all the bacteria in the water, and everything starts getting pumped up to the atmosphere.
If we look at reservoirs, and there's hydro dam reservoirs also on top of that, they are considered a methane source generation and carbon contributor to this category. What are the engineering solutions to that? How would you reduce their
No
emissions?
dams. No dams.
No, but okay, you're still going to have a dam. So what is it you do? You add more circulation of water? Do you
The problem with reservoirs.
engineered solutions?
Yeah, the problem with reservoirs and why they end up as huge methane sources is because you have a lot of organic matter coming with it, being transported with the river, and then it all ends up stuck at the dam site. So you have this accumulation of organic matter coming in and being stored in the reservoir, and then this creates a high biological oxygen demand because it has to be decomposed, which completely ends up using up all the oxygen in the water. You have anoxic conditions, hence you have methane production, right? So if we wanna keep the dam and sort of limit methane production, I can think of several things. Either you start building up some facilities on the way in the rivers to bring less nutrients, take it up before it ends up in the reservoir. Or you can also, depending on the river ecosystem, depending on biodiversity, of all these considerations have to go in, you can make the water pass through certain treatment facilities where you put, for instance, something like duckweed, which takes up a lot of phosphorus and nutrients from the water system. So it automatically clears the water. You can then remove the duckweed, use it as biofuel or whatever else. And then you end up with a much clearer water coming into the reservoir. It's not going to completely stop all methane emission, but it's going to probably reduce it. The other aspect is you might figure out some engineering mechanisms as to how to limit disturbance. Because with methane emissions, the more the soil is disturbed, especially with ebullition, you can probably see this if you go to a lake, which has a lot of methane stored. If you start stepping on some of the soil, you start seeing bubbles. Because the more you disturb the soil, the more of these methane will be emitted by ebullition. So with the whole hydropower thing, with the dam, with the engineering, if you can figure out some ways to disturb the ways where the sediment might get disturbed, that will also decrease the methane ebullition. With methane ebullition, it's worse than the dissolved methane because when the methane is being dissolved into the water column, especially if you have a large depth, it might get oxygenated on the way and gets broken down into CO2. So CO2 is, as a greenhouse gas, it's better than methane. But with an ebullition in a bubble, it rises up so fast and it doesn't get mixed in the water, nothing happens. The whole methane which gets released from the sediment ends up in the atmosphere. So that's another possibility. At the time being, I can think of those two solutions. But
they make for beautiful photographs when they're frozen. Yeah.
Sure. Yeah.
And there's no way of, no technology, that's for harvesting that underwater.
Well, for methane, there are some techniques. I'm not an engineer, but I've read some papers about extracting the methane that's being produced under the sediment, if you have a very thick sediment. But most of these engineering projects are extremely expensive, and we're talking about millions of euros. So I don't know if...
Hypothetically.
Yeah, some of them. So I don't know how much it makes sense to, for small dams, for small hydropowers, it doesn't make sense to do. If you have a massive dam, it might be worth investigating. But in general, we're still not there yet, I think.
Lake Sevan duckweed,
No.
is it a solution? No?
No. Not in Lake Sevan. It might be a solution in the tributaries ending up in. So if you have all the, for instance, wastewater or rivers bringing up a lot of nutrients, you do it in the tributary before it ends up in the lake. And it's such a huge lake. First of all, I'm not sure if it's feasible. Second of all, if you put duckweed in the water column, you don't get light penetration. It kills a lot of the benthic production. So it might be a solution to pass some of the very dirty tributaries through a duckweed farm. Before it ends up in the water, you get cleaner water into the lake, but not in the lake, for sure.
I didn't see any duckweeds in around Lake Sevan watershed. But a lot of duckweed built up is going on in summertime in Ararat Valley, in the canals and small ponds,
That's
a lot.
yeah, quite likely it happens a lot.
Sure.
Yes.
Most of the time you are talking about the total carbon
Yeah.
and total phosphorus.
Yes.
But do you also have any data about the bioavailability of these elements, like stable and labile forms of the elements?
With carbon, less so. We did look into a bit, but we didn't go so much into the quality of the carbon, into the DOC. With phosphorus, yes, we also measured SRP, the soluble reactive phosphorus, in most of our studies. The problem with soluble reactive phosphorus is it gets taken up very quickly.
Right.
So in most aquatic ecosystems, you have some kind of release of SRP, and then if you're going and measuring it once a week or once a month even sometimes, you might lose these peaks
Right.
because they're taken up immediately by the algae, right? So when they're taken up by the algae, you can still measure them in TP, total, but unless you have a very frequent measurement, sometimes SRP is, well,
Because it will give you an idea about the age of
it
the...
does. I didn't show here, but in the publications of these papers, we show the SRP as well. So if you want, we can talk more in detail afterwards, and
Sure.
I can show you the graphs, but I don't know if I have.
If you check the monoesters and diesters percentage of total phosphorus, it will give you the age of the soil. So in that case, you
The
can...
phosphorus in the sediment? Or
Yeah, in the soil also,
I don't know.
even in the water column.
Yeah, that's possible actually. We haven't looked at it for these studies, but yes.
Okay.
Thank you first for your nice presentation. I probably come with a more diverse background like yours. By profession, I'm also having a PhD in marine science, and I'm more of a microbiologist. So what I find during the whole talk, it's all about carbon cycling. And I believe, and I think others would agree as well, the biggest role is being played by the microbes, who transforms different forms in between. Your cycling, your mineralization, releasing them back, plants taking it, getting it released, bacteria working upon them. So did you have a more data on those kind of stuff, or I can get them from your publications?
Yes. Well, the first project specifically, I had a colleague, a couple of postdocs actually, that looked into microbial mineralization, both in the water column and in the sediment. In the sediment, we saw that mineralization was very low, as I showed, because it was mostly anoxic. In the water column, they measured both mineralization rates, and they also measured the source of the carbon. One of the surprising results that we weren't expecting in such systems that get a lot of carbon and nutrients from the surrounding was we thought the majority of the carbon being mineralized in the water column was supposed to be, or naturally from allochthonous, from terrestrial sources coming into. But then what she found out, my colleague, was that the majority of the mineralization happening was from internal sources. So the algae, the phytoplankton, the periphyton, and so on, was being mineralized very quickly. That might go into this labile versus recalcitrant carbon dilemma or issue. If maybe some of the terrestrial sources that are coming in are harder to degrade by the bacteria versus the already available algae, the carbon, the organic material, which might be easier. We did look into it. I'm not a microbial expert, so but I can point you to all the publications from these projects, and you can probably look into more of them.
I saw you referred to Lars' paper, 2009, Limnology and Oceanography. I worked with Lars during that time. And in those schedules, I mean, that was my next question coming, that whether you did look for the sources of the carbon, whether they were autochthonous or allochthonous. So you said your colleagues had looked into that and they were mostly autochthonous, not allochthonous.
Yes, at least throughout the summer. So basically, our conclusion was that throughout the majority of spring, summer, the majority of the material that was mineralized in the water column was autochthonous, internal loaded carbon. The allochthonous sources were mostly coming in with snowmelt.
Yeah, exactly, because you weren't expecting the allochthonous coming during the summer months because you don't have the runoff coming into the head level or even the lakes.
Yeah. So basically we had some peaks of nutrients and also carbon around March when the ice and snow starts melting from the nearby fields. You get a lot of these ice melts ending up in the kettle, it gets flooded a bit and the nutrients increase. But throughout the rest of the year when we had high production, the majority of the mineralization was autochthonous. Which was, quite frankly, like I said, a bit of a surprise to us because our initial hypothesis was that the majority is external carbon. But
while doing science, let's not get surprised. We reveal everyday new stuff. You come from Germany, you said. You studied in Germany.
I come from Lebanon. I did my PhD in Germany and I worked there.
Yesterday, 4th of February, there is a paper from AWI, Alpha Institute of Limnology and Polar Biology. They published a paper. We had all these years thought of a lot of carbon getting stuck into the permafrost of especially the Canada, Siberia, and even Russia. And they have found that when it's the shallow water along the continental shelf, there are some ditches kind of structure where they are very less available to the microbes to work on, even though we have microbes in the permafrost area. But that has changed a little bit of scenario what we have been thinking all these years, that everything is stored there in the permafrost, but a lot of them are being remineralized and getting back into the environment. And that becomes a problem given the situation with the climate changing now and global warming, which many people are reluctant to believe. In the last 5 years I was working in the States, where a big group of people don't believe that, including our president, that there is no climate change. But this has been happening.
Well, from my experience, it's usually not just the fact that it doesn't happen versus it happens or it starts to happen. It's also all about the rates, right? So you might have some mineralization, you might also have some bacteria in the permafrost, but then what happens when it melts? You still have some mineralization, but the rate picks up, right? And then you have to look into what difference does this change in rate trickle down into the overall ecosystem or global issues. So it's a very similar scenario in those freshwater systems, that you have a lot of the processes that happen all the time. But then what happens with temperature increase, what happens with changes in stable states and so on, is that those rates change drastically and significantly. And when that happens, most of the time, because everything is linked, you have this domino effect that ends up changing emissions, ends up changing food webs and so on. And a lot of time they have very important repercussions.
Just one last thing. Dr. American asked you about if you have any plan to, if you're asked to, let's say, dam you have where you have lots of CH4, that's methane generation. You talked about duckweed, but I don't know if it is feasible in small streams of Armenia or the rivers, which are mostly running through the highs and lows of the mountains, to grow duckweed in such a uneven bottom and also flowing water. Or if we could think of making some kind of fall where we could have a water flowing from the top and heavier particles settling down, and then that way you could reduce the inflow of organic matter into your dam.
Well, with flowing water, it depends how fast it's flowing. If it's fast flowing water, you cannot have duckweed. Duckweed only grows in more or less stagnant water. So if you do some sort of engineering process to have duckweed farms, you have to make canals where the water travels or flows very slowly, right? So this is part of the engineering process. The other process with the sedimentation and increasing sedimentation, this has been done before in rivers, where you build certain mechanisms to capture the bigger particles. Again, it depends on the budget. Usually these stuff cost. Yeah, it depends on the size of the dam as well, if it's worth making all these, and the size of the reservoir most of it, because that's proportional to the emissions. So if you have a big reservoir, if you have a lot of money, a lot of these processes are possible. And then you can collect the duckweed and use it as biofuel, source of energy, or feed for some animals or whatever.
So more of a fodder than biofuel maybe.
Yeah, but it also depends on the streams, right? If you have some streams which are important for biodiversity, important for different habitats as for animals and so on, you don't want to engineer the whole thing and make it artificial. So the answer I gave is quite general, but then you have to look into the project itself, the habitat, the system, and then plan something which fits for most animals.
Okay, well,
Thank
thank
you
you very much. Okay, thank you very much. No other questions, I suppose? Yeah? Gara, this was a great talk. Thank you very much for taking the time,
Thank you for having
presenting
me.
it to us, and look forward to being in touch a lot. Thank you.